Oops. All right. Should I start?
Yeah, sure. Please go ahead.
All right. Good morning to everyone from Athens. I'm Eleni Anyfandi, clinical neuropsychologist and researcher in National Kapodistrian University of Athens in the Laboratory of Experimental Psychology and Neuropsychology. Very happy being here presenting my research. I will present to you about chronic lithium and valproate effects on cognitive flexibility. And I will try to present to you translational study of our lab. Starting with, I would like to state two questions that will be answered through this presentation to give a background. I will say, can we consider lithium as a mood stabilizer exclusively? Or can we use it in another way? Well, for decades, lithium was considered as a mood stabilizer only, either used as monotherapy or an add-on. But in the later years, somehow recently, let's say the 10 years now, there was a twist in that notion. So many researchers had started to imply more and more frequently that lithium can be beneficial in other neurodegenerative disorders, such as dementias. What we do know about lithium, the extensive use in everyday psychiatric clinical life, are substantial to investigate lithium effects on cognition. Well, what do we know about the neurotrophic and neuroprotective effects of lithium? There are so many researches showing the neuroprotective and neurotrophic effects of lithium. About the neurotrophic action, it seems that we have increased cell proliferation, increased cell differentiation, increased neural growth and regeneration, enhancement of action formation, that means plasticity. And in the neuroprotective field, we see that lithium can inhibit or decelerate, let's say, apoptotic mechanism and cell death that is caused by acutely caused damage. And the neurotrophic neuroprotective mechanism of lithium are based on inhibition, mostly on inhibition of apoptotic kinase GSK3B, and lithium affects kinase GSK3B directly and indirectly. And in that way, through GSK3 inhibition, it affects activation of cell survival pathways, such as PI3 kinase with ACT and Wnt with B-catenin, or MEK-ERK pathway. It also induces survival molecules, such as anti-apoptotic BCL2 protein, but mostly affects BDNF, brain-derived neurotrophic factor, vascular endothelial growth factor, VEGF, and glial-derived neurotrophic factor, GDNF. It also enhances autophagy, removing proteins that accumulate in the neurodegenerative diseases, and that is something we have seen through knockout genes rat models, such as rat models of Alzheimer's disease or TBI's models. It also inhibits through NMDA excitatory receptors, and it reduces induced excitotoxicity. Well, the other big mood stabilizer that is used in everyday life is valproic acid. Valproic acid also has neurotrophic and neuroprotective effects, and it's also widely used as a mood stabilizer, as a monotherapy, or in combination with lithium or other anticonvulsants, antipsychotics, and antidepressants. VPA can enhance mostly gamma-aminobutyric acid, GABA, neurotransmission and the inhibition of enzymes involved in GABA metabolism. But the main effect of VPA is the inhibition of histone deacetylases at therapeutic serum levels. So the two big mood stabilizing drugs, lithium and VPA, have different pathways, but have the same behavioral and biochemical targets. Lithium through GSK3 mostly, and valproic acid through HDACs, target at the end neuroprotection, neurotrophism, as we've seen through BDNF and GDNF, anti-inflammation, something I didn't mention before, but it does, they do reduce interleukins 6 and 10. They induce neurogenesis and angiogenesis through VGNF. They regulate microRNA. They do, they perform stem cell migration. They do mood stabilization in behavioral field, and they have antidepressant, anti-anxiety effects. So both lithium and VPA have strong neurotrophic, neuroprotective effects, mostly lithium, if we can say, and they're both used in mood stabilizing. But regarding lithium, there is something more. There is so many literature saying that lithium and its neuroprotective and neurotrophic action maybe can be used in other neurodegenerative diseases such as Alzheimer's or TBI or stroke, or so many other neurodegenerative diseases such as Huntington, Parkinson's, Fragile X, and so on. But if we see the whole literature, then we conclude that we can surely conclude that neuroprotection is the most constant result of lithium usage. And I can say there are even more researches about the neuroprotection than its clinical use. And if we see the whole literature, then we will see that even bad clinical responders in lithium have neuroprotective, produce neuroprotective effects, either or on neurochemical or or biological base. But someone can ask, is neuroprotection constant over time? So if I get lithium and then I stop it, will it protect my cells in the same way? Research findings supports that neuroprotective effects of lithium are expanding over time. Some neuroimaging studies and some more have presented that the association between lithium treatment and hippocampal, let's say, volume seems to be independent of long-term treatment response, even for patients that have been exposed to lithium in the past. So hippocampal volumes are larger and better than patients not exposed in the lithium, even patients have been exposed in the past. Consequently, these effects of lithium on brain structure may be generalized to patients with other neuropsychiatric illnesses other than bipolar disorder that's mostly used. But the big question, let's say, that is, can this effect be translated into a cognitive benefit? Because we know that we can see neuroprotection in some treatment drugs, but that can't be translated into cognitive benefits. And this is a big a question about this new drug, aducanumab, that protects from cell death and reduces the tau protein accumulation, but we are not sure if it does cognitive benefit on patients with Alzheimer's disease. In our lab, we deal with the investigation of functional and cognitive effects of lithium and chronic administration of lithium. What we do know about the relevant clinical but not experimental literature is that in the past, previously suggested literature was saying that lithium has had a negative effect on memory. Studies from our lab and now on from other labs, but studies from our lab were the first that reported functional improvement of cognitive performance as a consequence of long-term administration of low doses of lithium. Based on those findings, we were asked to review a special issue of Psychopharmacology journal, Cognitive Enhancers, to give our knowledge about lithium. And then the question is, how we will investigate cognitive effect of lithium? Lithium we know that it's approved as a mood stabilizing agent, indicated as monotherapy for treatment of bipolar disorder. It can treat acute manic or mixed episodes, and it can be used as maintenance treatment. So bipolar disorder patient is the only clinical group that we can use for investigating lithium's effect on cognition. So then we must know what about cognition in bipolar disorder. Though there is a significant clinical and experimental interest in the impact of bipolar disorder on cognition, especially in executive processes, bibliography has produced frothy and inconsistent results. These recent reviews and this difficulty is attributed to methodological variability mostly across studies, poor characterization of clinical samples, and the difficulty, this is the most difficult part, separating the cognitive impact of bipolar disorder from that of pharmacological intervention used for the relief. What we do also know is that there are differences among BDI types, BD type 1 and BDI 2 in cognitive deficits, where BDI type 1 demonstrate impairment in verbal fluency, working memory, attention, set shifting, inhibition, and planning mostly. And BDI type 2 patients demonstrate lesser but not less worse deficits in cognition, such as verbal fluency, working memory, and attention. Cognitive flexibility, which is here the target cognitive function, and set shifting, which both play a fundamental role in executive function, has been poorly investigated. And what about pharmacological effect on cognition in bipolar disorder? The impact of pharmacological treatments upon cognitive function is mixed. A number of studies reporting conflicting results. There are evidence in generally that lithium may improve some cognitive domains, but also it can impede others. It's sure that antipsychotics may be detrimental to cognition. Valproic acid, it seems that also associated with worsening cognition. Overall, quality of evidence is poor and is derived from a relatively small number of studies, methodologically heterogeneous. In conclusion, there are strong evidences that lithium and VPA can be beneficial, with lithium, let's say, taking a strong advantage over VPA to do its biological mechanism. It is difficult separating the cognitive impact of bipolar disorder from that of the pharmacological intervention used for its relief. Cognitive flexibility has been poorly investigated. So we propose a methodological way to disentangle cognitive effects of lithium of those caused by bipolar disorder, and that is a translational study. So in our study, we have two parts, a clinical part and experimental part. And in the whole, this is a two-folded research combining clinical and experimental procedure. Investigation of cognitive flexibility, which was our target here, was assessed by intra-extra dimensional shift ID of Cambridge Neuropsychological Test Automated Battery, CANDAB, which is an analog of Wisconsin card sorting test, where there are visual discriminative stimuli, for example, pink shapes and white lines, and this is part A. For the experimental part, we used an animal model assessing set shifting produced from Birrell and Brown that is almost identical to ID CANDAB, where odor and texture for rats are discriminative stimuli. And for example, we used paper and cinnamon, paper for texture and cinnamon for odor. And this is part B. For both parts, we measured stage errors. How many errors did the subject have done to change the stage? So in our clinical study, we used two euthymic bipolar disorder patient groups, psychometrically masked for on Young Mania Rating Scale, Hamilton Depression Scale, mini mental state of examination, and were demographically matched healthy controls assessed with CANDAB ID. The pharmacological group, the treatment, were treated with lithium, either with lithium or valproic acid over 2 years, that is chronic administration. The group were as follows. We used controls, bipolar disorder patients treated with chronic lithium, and bipolar disorder patients treated with chronic VPA. In our experimental study, we used 5 groups of male Wistar rats matched for age and adlibit body weight in set shifting rodent model. The pharmacological groups were treated for 2 months with intraperitoneal injections of either lithium or VPA. Here we see that we have identical groups with the clinical groups on rats, but we have also add 2 more groups. We have add acute lithium administration to see what's the effect of acute lithium administration, and we have add chronic administration in the past to ask if there was a significant effect of lithium long-lasting after the treatment. Here we see the ID and set shifting stages explained. I won't stay that slide. I will proceed to our results. These two slides, in the next two slides, I present the clinical and experimental results. What we can see in both graphs, the clinical and the experimental graphs, is that clinical groups do not differ between them. So let's discuss the results. The lithium and valproic acid both bipolar disorder groups on the clinical study were indistinguishable in psychometric performance. Young Mania Rating Scale, Hamilton Depression Scale, Mini Mental State of Examination scores within non-clinical range. It seems that our groups were very well characterized clinically and comparable in clinical history. Percentage of bipolar disorder types, hyperthymic and depressive episodes, hospitalization, adjunct antidepressant, antipsychotics. There was no difference in performance of the clinical groups from controls in cognitive flexibility as assessed by CANDAP ID. Here I will add some more info. In contrast, in the same study, we're preparing that study, both bipolar disorder groups performed significantly worse than controls in CADAP PAL, which assesses memory, SST, which assesses inhibition, and CGT, which assesses risk adjustment and risk taking, with minor deficits in SOC, which assesses problem solving and strategy. The analogous animal study failed to demonstrate the deleterious or beneficial effects on set shifting of chronic ongoing exposures to VPA or lithium or chronic exposure to Li in the past with the rats. The only deficit observed was increased response latencies after acute lithium exposure, unaccompanied by increased choice errors, a finding suggesting that transient psychomotor retardation after lithium acute treatment. These results are congruent with our earlier reports of no effect of chronic lithium on attention, let's say latent inhibition in the rat, along with beneficial effect on this lithium regimen on working memory and long-term fear retention, provided that the demands of these tasks extended to the verge of the cognitive capacity of unmedicated rats. Conclusion. In conclusion, chronic lithium and valproic acid, which offered no protection to basic cognitive functions such as spatial learning and memory and response inhibition in euthymic bipolar patients, accompanied with normal executive performance of ID. Within the limitation of the translational approach, the combined results suggest that normal cognitive flexibility in euthymic bipolar patients is not attributable to a direct beneficial effect of the two stabilizer on the target task. It is also noteworthy that the normal ID performance was manifested in spite of congruent pronounced deficit in spatial learning and memory and milder ones in the response inhibition. I must say that this work was co-founded by the European Union and Greek national funds through the operational program Human Resources Development, Education and Lifelong Learning, under the call supporting researchers with an emphasis on young researchers, Psycho B. Thank you.